Immunology Insights, Basics and Beyond, Chapter 42, Tumor Immunology Overview. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we honored Sir Peter Medawar, a true pioneer who helped us understand immune tolerance and its pivotal role in transplantation. Today, we begin a brand new chapter in our immunology journey, cancer immunology. And to start, we will take an in-depth look at tumor immunology. Cancer is one of the most challenging and devastating diseases in medicine, affecting millions worldwide and capable of arising in virtually any tissue. It is not only a major cause of death, but also a moving target, constantly adapting and resisting our best therapeutic strategies. Understanding how the immune system interacts with tumors is essential for grasping how cancers develop, progress, and in some cases can be defeated. This section will guide us through the ways the immune system recognizes and combats tumors, why it sometimes fails, and how these insights have led to groundbreaking therapies like immune checkpoint inhibitors and CAR T-cell treatments. So let's talk science. What is tumor immunology? Well, tumor immunology is the study of how the immune system detects, responds to, and sometimes fails to control cancer cells. Unlike viruses or bacteria, tumors arise from the body's own tissues, making them difficult to distinguish from healthy cells. Despite that, the immune system has a surveillance system in place, known as immune surveillance, to catch early signs of cellular transformation. This system is constantly on the lookout for cells that divide abnormally, accumulate mutations, or display danger signals. When successful, immune surveillance can destroy these cells before they develop into tumors. But when the cancer escapes this watchful eye or actively suppresses the immune response, it can grow unchecked. Let's reiterate the basics of immune surveillance. Meet the key players involved in the immune system's cancer patrol. First, natural killer cells, or NK cells. Part of the innate immune system, NK cells target abnormal cells with low or missing MHC class I molecules, a common feature in tumor cells trying to avoid detection. Second, cytotoxic T lymphocytes, or CTLs. These adaptive immune cells recognize tumor-specific or tumor-associated antigens presented on MHC class I. Once activated, CTLs destroy the target using cytotoxic granules like perforin and granzymes. Third, dendritic cells, or DCs. These antigen-presenting cells are responsible for picking up tumor antigens, migrating to lymph nodes, and activating naive T cells. And fourth, macrophages. Tumor-associated macrophages, or TAMs, can be friend or foe. Some attack the tumor, but many adapt to support tumor growth, suppress T cell responses, and promote blood vessel formation. Together, these immune cells form the foundation of the body's anti-tumor defense. But unfortunately, tumors don't go down without a fight. So, what makes cancer cells so hard to kill? Cancer cells are tricky. Since they originate from normal tissue, the immune system must walk a tightrope, be aggressive enough to detect abnormal behavior, but tolerant enough to avoid attacking healthy cells. Here are some of the tactics tumors use to survive. First, immune tolerance to self. Because tumors express many normal self-antigens, the immune system often ignores them. This limits the natural ability to mount a strong immune response. Second, antigen loss variants. As cancer progresses, cells that express recognizable antigens may be killed off, while others with mutations that hide the antigens survive and proliferate. Third, an immunosuppressive microenvironment. Tumors actively shape their surroundings. They release TGF-beta and IL-10, recruit regulatory T cells or Tregs, and myeloid-derived suppressor cells or MDSCs, and create hypoxic acidic conditions that shut down effective immunity. And fourth, checkpoint molecules. Tumors frequently upregulate PDL1, which binds to PD1 on T cells, essentially switching them off. This mechanism will be discussed in depth when we talk about checkpoint inhibitors. These immune escape strategies are central to why cancer can grow, spread, and sometimes resist treatment, even when the immune system is fully active. Now, let's look at tumor-associated antigens or TAAs. TAAs are molecular fingerprints that the immune system can potentially recognize and target, even if the tumor is self-derived. They fall into three main categories. First, mutated self-proteins. Alterations in oncogenes or tumor suppressor genes like p53 or KRAS can produce abnormal protein structures recognized as foreign. Second, cancer testis antigens. Normally restricted to reproductive tissues and silent in healthy somatic cells, these proteins, for example NY-ESO-1 or MAGE-A, are apparently reactivated in tumors and often trigger strong immune responses. And third, viral antigens. In virus-driven cancers, the tumor expresses viral proteins like E6, E7 from HPV in cervical cancer or LIMP1 from EBV in certain lymphomas that can serve as clear non-self targets. Studying TAAs is crucial because they form the basis for cancer vaccines, monoclonal antibody therapies, and engineered T-cell approaches. Now, let's assess the clinical relevance of tumor immunology. Well, tumor immunology is not just about understanding disease. It directly shapes prevention, diagnosis, and therapy. Let's look into the four main areas where this knowledge makes a difference. First, diagnosis and prognosis. Immune biomarkers such as PDL1 and MSI, which means microsatellite instability, help predict patient outcomes and guide treatment selection, particularly for immune checkpoint inhibitors. PDL1 is a protein expressed on tumor cells that can bind to PD1 receptors on T cells, dampening their activity. Its presence can indicate whether blocking this pathway might restore anti-tumor immunity. MSI reflects defects in DNA mismatch repair and is associated with high tumor mutational burden, which often makes tumors more visible to the immune system and more responsive to immunotherapy. Another key prognostic factor is the presence of tumor-infiltrating lymphocytes or TILs, immune cells, mainly cytotoxic T cells, that have migrated into the tumor microenvironment. High TIL levels often indicate an active anti-tumor immune response and are linked to better survival in cancer, such as melanoma and certain breast cancers. Conversely, low TIL numbers may point to a more immunosuppressive tumor environment and poorer prognosis. Second, treatment innovation. Immunotherapies have joined surgery, radiation, and chemotherapy as the fourth pillar of cancer treatment. Checkpoint inhibitors can release the brakes on T cells, while CAR T cells and bispecific antibodies directly target tumor antigens, offering new hope in otherwise treatment-resistant cancers. Third, prevention. Vaccines such as the HPV vaccine and hepatitis B vaccine can prevent cancers linked to these viruses, showing that immunology can be proactive and not just reactive. And fourth, monitoring. Advances like liquid biopsies, so detecting tumor DNA and immune markers in the blood, enable ongoing, minimally invasive monitoring of both disease burden and treatment response, allowing therapy to be adapted in real time. To conclude, tumor immunology teaches us that cancer is not just uncontrolled cell growth. It's a high stakes duel between mutation driven disease and immune defense. Consequently, it is not only a biological problem, but also an immunological one. The immune system is both a vigilant gatekeeper and a potential executioner. When it works, it can eliminate cancer before symptoms appear. When it fails, tumors exploit that weakness to grow, spread, and evade detection. Understanding these immune-tumor interactions is key to modern oncology. It drives the development of highly personalized therapies, smarter prevention strategies, and earlier detection tools, while helping us select the right patients and anticipate resistance. Most importantly, it offers hope because by harnessing the immune system, we can fight cancer with unprecedented precision. In the next chapter, we will explore immunoediting and immune evasion, the evolutionary arms race between tumors and the immune system, where each continually adapts to the other's moves. Basically, a dynamic battle between mutation and immunity. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.